All right, you guys, I am back here to finish this off with you. And we're going to talk in this video about blood flow. So we've talked about cardiac output. We've talked about fluid volume. We've talked about ways to control the blood volume, pardon me. And now we want to talk about how we can control the way blood flows through the body, keeping in mind that you only have 5 liters of blood, give or take. And therefore, being circulated, right? Your cardiac output is roughly around 5 liters, 5.5, 6, somewhere, depending on some variables. So it doesn't make any sense to think that every blood vessel you have has blood in it at any given time, because there's just not enough. So what we have to understand is that your cardiac output is distributed unequally from throughout different organs due to what's happening in terms of resistance to blood flow through those organs. So we're going to talk about blood flow, and we're going to talk about factors that alter resistance. Resistance just is anything that impedes the flow. So the higher the resistance, the lower the flow. So if we're trying to control how blood flows through the body, the best way we can do that is control the resistance in certain areas, because that would decrease flow in that area and then send it someplace else. All right, so you don't need to memorize any of these numbers, but just for an idea, you can see how your blood is kind of distributed at any given moment. So if you had 100 mils of fluid in a minute moving around, about 24% of that blood would be in the gut and liver. 19% would be in the kidneys, 13% the brain, 4% the coronary arteries, the 21% the skeletal muscles, 9% the skin, and then the other remaining 10% is going to be distributed across other organs depending on demand. Okay. Sorry, I was going to say I'm going to sneeze. Too late. Okay, so let's think about this. So what we're looking at now is how is blood flow controlled? So there's some laws, of course, that govern how blood flows. One thing for sure that we need to understand is blood is going to flow from a region of higher pressure to lower pressure, high to low. That makes sense. That's a gradient. Gradients allow for things to move. So this is a pressure gradient. Blood will flow from a region of high pressure to a region of low pressure. The rate at which blood will flow is going to be proportional to the differences in pressures, meaning high pressure, high flow, low pressure, low flow. So we can say the pressure and flow are proportional. Pressure up, flow up. The rate of blood flow is then inversely proportional, opposite directions, to the frictional resistance to blood flow within the vessels. So that can be illustrated here. So we have blood flow is inversely related to resistance. So this is the change in pressure. So we could say the pressure difference between the two ends of the tubes, right, through the circulatory system. So blood flow equals the change in pressure divided by the resistance. So pressure and flow are directly related. So let me write that here. Blood flow is directly related to pressure. This is like watching a kindergartner, right? Blood flow is inversely related to resistance. The more resistance, the lower the flow. The lower the resistance, the higher the flow. Okay, let's keep going. The picture. So we're talking about the human. So we know that the pressure is highest. So this is your heart in this little box. This is your pump. This is the left side of the pump. This is the right side of the pump. The left side of the pump is going to pump blood against the highest pressure you're going to find anywhere in your body. So when the blood leaves the left ventricle, it's leaving at a pressure of somewhere in the neighborhood around, just nice round numbers, about 100 millimeters of mercury as we go into the arteries. So there's your mean pressure in your arterial system, 100 millimeters of mercury. When the blood gets all the way back to the right side of the heart, the pressure is zip, very little. So that's a pressure gradient. Your pressure gradient is the difference. So your change in pressure right there is the difference between the pressure here and the pressure here. So if we are leaving the heart, left side of the heart at 100 millimeters of mercury, and we're coming back at 0 millimeters of mercury, the difference between 100 and 0 is 100. That's a pressure gradient. Remember, pressure is the force exerted by the blood on the walls of the vessels. For blood to flow, there must be a pressure gradient. And I mentioned this already. Increase in pressure, I'll write it again. Increase in pressure pressure is going to mean we're going to have an increase in flow. So what kinds of things affect pressure? Remember, pressure is the force exerted by the blood on the walls of the vessels. So the more blood you put in there, the more pressure you're going to have. So things that we've talked about so far, things that are going to affect pressure, anything that's going to affect cardiac output, right? That's the blood going in there. So your heart rate and the other variable for cardiac output, if you remember, was stroke volume. Remember, CO equals stroke volume times heart rate. So any one of those variables is going to increase pressure or affect pressure, I should say. Okay, so that's pressure. A pressure gradient is necessary. There's some other things that are going to affect flow, but really what they're going to do is they're going to affect the resistance, right? We'll talk quite a bit about resistance in a minute. So resistance can be measured using a couple of variables. So we're going to look at some things that affect the blood flow. One is the length. So here's our equation right here. This is what this equation for resistance means. The length of the vessel multiplied by the thickness of the fluid, the viscosity of the fluid, divided by the radius to the fourth power. Now, I'm going to start, I'll talk about each of these independently in a minute, but of those variables, the only one that really is modifiable in a moment-to-moment way is the radius. The length of our vessel and the viscosity of our fluid do not normally vary from moment to moment. So they're changing those variables to affect blood flow in an instant is not going to work. Okay, what else do we need to know here? So the two things that we're going to care a lot about are the radius in terms of the important factors for blood flow. The radius of the tube is a big one. It's to the fourth power. That's an exponential kind of a deal there. So that's something significant. And the pressure, mean arterial pressure, the pressure in the system. Those are the two things that are going to be the biggest players for us. And what that means is, in order for us to affect blood flow, we can either play around with the pressure or we have to play around with the resistance. And what we're going to learn is that the best way to play around with the resistance is to alter the radius of the tube. The radius is the diameter of the tube, the lumen of the tube. So I'm going to tell you this now, and I'll may say it many times. The vasoconstriction or vasodilation. Vasoconstriction means making the tube smaller in the inside. Vasodilation means making the tube bigger on the inside. Vasoconstriction of the arterioles, those are the only ones that can do it, provide the greatest resistance to blood flow. And using the diameter of the arterioles, or I should say modifying the diameter of the arterioles, is the way that we can direct blood flow from one area of the body to another. Send it away from a place we don't need it and send it towards a place we need it. Now, length is a variable. If you could make your vessels longer or shorter, that would affect fluid flow. If you're talking about a hose in your yard, the longer the hose, the lower the flow. The shorter the hose, the higher the flow, because length of the tube affects the resistance to flow. A long tube increases resistance and therefore would decrease flow. Now, your tubes are all connected to other tubes. So we have a closed circulatory system in our bodies. So getting our tubes longer or shorter aren't going to work for an immediate kind of change. Yeah, if you gained a bunch of weight and you had to grow longer vessels over a period of many years, that would affect resistance in your body to flow, your resistance to flow for your blood. But that's not going to give you more blood in your feet in right now, or it's not going to send blood to your brain if you need it, because it's just not modifiable. All right, same with viscosity. That's how thick your blood is. The thickness of your blood depends on what's in there. Mostly the red blood cells, because we have tons of them. White blood cells, if you have a lot of white blood cells, more than you should, that will change the thickness of your blood, but that usually means you have some other problem, like potentially an infection, or worst case scenario, some kind of a leukemia. But that could change the thickness of your blood. Changes in red blood cells, if you're anemic or you have polycythemia, remember we've said those terms before, that would change the thickness of your blood. If you had too many or not enough proteins in your blood, that would change the thickness. However, that's not what we use to alter blood flow in a moment-to-moment way. That's off the table as well. But you can see the relationship, and that makes sense. An increase in thickness increases the resistance. You've all had the opportunity, I'm sure, to drink out of a straw. Like if you try to suck up a milkshake, which is a very thick fluid, it's a lot harder to suck a milkshake up through a straw than it is to suck up some, soda water or soda or whatever else that would be thin. That would be the same, that would apply to your blood. But again, that's not going to be something you're going to change, in this minute to alter blood flow. That leaves us with the radius. This is the big player. The radius of the tube, the diameter of the tube, the diameter of your blood vessels is going to affect the resistance to flow. And this is the most important. And remember, this is what we're talking about here really are the arterioles. Those are the most important. You want to remember that. Small arteries as well. But the arterioles are where we're going to be able to regulate the radius of our tubes most efficiently. And this is a big deal. This is a fourth power effect, right? So that means that small changes in the radius of your vessel lead to large changes in resistance, right? So the smaller the tube, the larger the greater the resistance, and therefore the lower the flow, right? It's a fourth power effect. So that means, like, if for some reason, if you increased your radius of your tube by 2 millimeters, that would increase the flows essentially 16 times, right? That's a huge difference. Just doubling the size of your tube. It's a big, big change. All right, so that's the big one. So here's an idea. Here's a little picture that shows us, right? So if we have, you know, your radius is a millimeter, your blood flow is, you know, coming in this direction. The radius of that tube is a millimeter. The resistance is what it is, and the flow is what it is. Now, if we increase the radius of this tube, this one up here at the top, if we increase the radius here, we just make it, you know, from 1 millimeter to 2 millimeters. The resistance goes up 6 times. Or pardon me, we're making it bigger. The resistance goes down 6 times, and the blood flow goes up 6 times. If we make the tube half as big, the resistance goes up 6 times, and the flow goes down 6 times. 16. I'm saying. I hope I didn't say 6 before too. 16 times. It's a 4th power effect, right? So that's a big deal. That's a huge change in blood flow. So the take home here is if you increase the diameter of your tube, your blood flow goes way up. If you decrease the diameter of your tube, your blood flow goes way down. And it's a real significant effect. So what this means to us is, remember, the blood flow throughout the circulatory system is going to be dependent on pressure, which we've already talked about, and I'm going to show you another picture of in a minute, and resistance. And it's the resistance, the sum of all the vascular resistance in the entire systemic circulatory system that we want to know. And the sum of all the resistance in the vasculature is called total peripheral resistance. And I want you to remember that the arterioles are where this is all at, right? The arterioles provide the greatest resistance to blood flow and therefore allow us the ability to regulate blood flow significantly. So what else do I want to tell you? I don't know what that bullet meant. I put it in that bullet, greater than 60% of total. Oh, I think that means that your arterial diameter, is basically going to encompass about 60% of the total peripheral resistance in the system. That's just the diameter of your arterioles. The other thing we know is that we see the largest drop in pressure at the arterioles. And let me show you that on this picture right here. Okay, so here we are, the pressure. So here's the left ventricle, right? And we see that the blood being ejected from the left ventricle is being ejected at about 120 millimeters of mercury. And that pressure stays high in the other large arteries. And then it goes way down, right? And then we see this big drop. We see this big drop from as we move into the small arteries, and ultimately as we get into the arterioles and the capillaries. So we see this big drop when we go from the small arteries, arterioles, into the capillary beds, right? So it's like, somewhere from around 90 millimeters of mercury as we go into the smaller arteries, and we're like at about 40 as we go into the capillaries, maybe a little less even. 40, 30, somewhere between 35 and 40 as we go into the arterial end of the capillaries. That's a really big pressure drop. You don't see any pressure drop here. You see this big pressure drop here, a small pressure drop as we move through the capillaries, and then, then we're in our very low system where we have those high capacitance vessels. So where in the arterioles where we have all that smooth muscle and we have the ability to change the diameter, we call those vessels our resistance vessels, and we call the veins our capacitance vessels. Remember, I used that term in the other lecture. All right, so blood pressure now. Remember, the blood pressure is the pressure that's being exerted on the wall of the vessel where you're measuring it, and it's a result of how much on the force that the blood in that tube is exerting on the wall of the tube. So our blood pressure overall, if you're measuring your blood pressure, it's going to be dependent on how much blood is actually leaving the heart. And of course, we're going to multiply that. We're going to calculate that by knowing how much blood is ejected each beat and how many times the heart pumps in a minute. And we also need to know what's the peripheral resistance. What's the resistance to flow? So your blood pressure is going to be dependent on that. And the resistance is determined by the radius of the vessel. So your blood pressure, let's say your blood pressure is, 120 over 80, which is considered normal. That number at the top is 120 is the pressure that the pressure that the blood, the pressure, what am I trying to say? The pressure in basically in the vessel at the aorta, right? The pressure that your left ventricle has to generate to exert the blood. That's what I was trying to say. 120 millimeters of mercury. And then when the heart goes into relaxation, the pressure that remains is the pressure on the number on the bottom. So we oftentimes refer to this number as the systolic, and that's the diastolic. That's an easy way to kind of think about it. You could get a little bit more complex there, but I don't think we need to. The pulse pressure then would be the systolic number minus the diastolic number. That gives you the pulse pressure. Now, if you wanted to, so in that case, what is that, 40? The pulse pressure is 40. So then you can calculate your mean arterial pressure by taking the number on the bottom, the diastolic number, and adding 1/3 of the pulse pressure. So you would take 80, let's write it over here, to get the mean arterial pressure. It would be the diastolic number, which is the number on the bottom, 80 plus the 1/3 of the pulse pressure. So I said the pulse pressure was 40 divided by 3. That would give you your mean arterial pressure. I can't do that math right now because my brain is too busy thinking about what I'm saying. It's not hard math, but okay, that's the mean arterial pressure. All right, so what does that mean? What does that mean to all of us? So let's put it all together. All right, now what we're looking at here is how pressure gradients and resistance relate to flow. So we can say now, these are all variables we've talked about. We're just putting it together conceptually. Blood flow equals the change in pressure in the system divided by the resistance. So cardiac output is the flow. That's how much flow there is. Cardiac output equals then the pressure in the whole system, your mean arterial pressure, divided by your total peripheral resistance. So there you have it all dialed in. Your cardiac output is going to be dependent on the pressure in the system, the mean arterial pressure in the system, divided by the average resistance in the system. Right? So pressure gradients and resistance are really important because they affect cardiac output in a major way, right? We know that cardiac output and pressure would be directly related, mean arterial pressure. Cardiac output and an increase in total peripheral resistance would be inversely related, right? And so flow, that's the same thing, saying flow is the change in pressure divided by the resistance. So hopefully that makes sense to you guys. I think that should make sense. Just got to think about it for a minute. And, before we get into this last little bit here, why do we care about this? Well, we care about blood flow because blood is how we deliver our nutrients to our tissues, right? Without blood moving through the circulatory system, our tissues don't get any nutrients and we die. So that's why we care. It's important. It's a very important thing. We need to make sure that we have the right amount of blood flowing to all these areas. If we don't need that area, we don't want to put a bunch of blood there because that's just a waste of energy. So we need to be able to regulate where the blood is going. And so we kind of talked about, the way to do that, and that would be to either affect the pressure or affect the resistance in the tubes. But how that's regulated gets a tiny bit more complicated, right? It's just not completely haphazard and random. It's actually controlled. And so there's kind of two big ways we regulate blood flow. One of those ways, or one of those big headings, would be extrinsic regulation. And that's like a real big response where like you're using the nervous system, or you're using hormones or neurohormones to affect many, many areas in the body kind of at the same time. And that would be considered extrinsic. And the other option is intrinsic. Intrinsic regulation is kind of more driven by the actual need of the tissue. It's a little bit more local, I guess would be a good way to do that. With intrinsic regulation, we can use local communicating molecules, where we have molecules that are produced in one tissue, and those molecules that are produced in one tissue are controlling another tissue in that same organ. So it's short distance control. And then we have some metabolic control mechanisms as well. I'm going to walk you through these guys in a little bit of detail. All right, so let's talk about the sympathetic. So I'm going to go back for 1 second. So you notice I put sympathetic nervous system and epinephrine, and then in quotations or in parentheses, I put the parasympathetic nervous system. And the reason why I did that is because the parasympathetic nervous system really is not a big player in it doesn't affect the arterial diameter. So as a consequence of that, is not a big player in terms of the regulation of blood flow. But it does do something, and I'll talk to you about that in just 1 second. All right, so let me do this first. So let's look at the sympathetic nervous system. The sympathetic nervous system is going to communicate directly with the arterioles, because your smooth muscle that wraps around your arterioles have adrenergic receptors located there. So what we know is that in cases, when we see, an increase in cardiac output and an increase, we can increase cardiac output, and we can increase total peripheral resistance by the release of norepinephrine at the targets from the sympathetic postganglionic neuron. It's going to release norepinephrine. That norepinephrine is going to bind adrenergic receptors on the smooth muscles of our arterioles, in all of our viscera, leading near all of our viscera. Yeah, that's good enough. And in our skin, the arterioles that are taking blood to our skin, those locations have alpha adrenergic receptors. And when acetylcholine binds those alpha adrenergic receptors, we are going to see vasoconstriction. That's what happens. And when that happens, we're going to see an increase in total peripheral resistance and also an increase in pressure in the system. Because the fluid that's there, if you keep the same amount of fluid there and you make the tube smaller, the fluid that's in there is going to push harder on the walls. Okay. Let's see. What else do I want to say about that? Okay. Let's talk about parasympathetic nervous system for a second. The parasympathetic nervous system is going to release, the postganglionics are going to release acetylcholine. And the effects of acetylcholine in the vasculature are really limited to the digestive tract, the external genitalia, and the salivary glands. And what we see here is a little bit of vasodilation. It's really minor, minimal, and it's so minimal that it really doesn't do much to actually altering total peripheral resistance. It really doesn't do a lot. The sympathetic nervous system is the one that's going to be the big player in terms of increasing or decreasing total peripheral resistance. Okay, it does get a little bit more complicated than that, but I think that's enough for us right now. That's good. Epinephrine is going to act just like the sympathetic nervous system, of course, because epinephrine is going to be released by the adrenal medulla in response to sympathetic stimulation, but it's going to be released into the blood, and it's going to float around, and it's going to bind receptors. The beta receptors are a little bit more, they like epi a little bit more than the alpha receptors. The alpha receptors have a higher affinity for norepinephrine. The beta receptors have a higher affinity for epinephrine. The effects are a little bit different. Where we see this effect most predominantly is in the skeletal muscles, in the heart muscle, the vascular beds are in the heart muscle, and a couple other places, but those are the places that are important for us right now. And what happens when epinephrine is released into the blood at those locations I just mentioned, we see a vasodilation. And that causes a decrease in resistance at those locations. And that can, therefore, increase blood flow to those areas. So this is a fight or flight deal, right? This is when you're, theoretically running for your life, and you need to make sure that you're getting enough blood delivered to those tissues that you're going to need to survive. The skeletal muscles are one of them. Your heart is another one. At the same time, in the non-essential locations of the body, we're going to see vasoconstriction. So we see vasodilation at the essential places and vasoconstriction other places that are not important. So because of that, the overall effect on total peripheral resistance is either we maintain TPR or it increases. It depends on how much vasoconstriction you get. The dominant effect of epi is vasoconstriction, just like norepi. But you do see some vasodilation in certain areas with epinephrine. I lied to you a little bit. You will also see some vasodilation in certain areas with the sympathetic nervous system, but it gets way more complicated than we've really gotten into. And so I don't really necessarily think we need to do that right now. But if you just think about it logically, if your body with fight or flight vasoconstricted everything, that would be bad. Your blood pressure would go through the roof and you wouldn't get any more blood anywhere that you need it. The whole reason for the fight or flight response is to get blood where you need it, when you need it most. The same thing with exercise, right? Exercise causes sympathetic stimulation. When you're exercising, the blood is going to your skeletal muscles. It's going to your heart. It's not the time to go and to send a bunch of blood to your gut, right? That's why you're told by your parents when you're little, don't go swimming or exercising don't swim right after you finished exercising, or don't. Nothing to do with one another. Don't eat right after you finish either swimming or doing a lot of exercise, right? Swimming is the one that I was told all the time. You don't, right when you're like, don't eat a bunch of food and then go swimming, or don't, immediately after swimming, eat a bunch of food, because when you're exercising, swimming takes a lot of energy. When you're doing that, the blood is not going to your gut. You're not going to digest anything adequately. Okay, so the dominant effect of epi is the same as norepinephrine, and that is vasoconstriction. Okay, the last couple things I want to tell you. So that's that type of control when your nervous system is sending out these neurotransmitters all over the place, or your adrenal medulla is sending epinephrine out into your blood and it's floating around everywhere. That's extrinsic. That's not local tissue controlling local tissue. That's your nervous system controlling your body, essentially. So the last couple things I want to mention is what intrinsic regulation is all about. So intrinsic regulation is basically when the blood, the alteration in blood flow is driven by changes in tissue changes as a result of alterations in metabolic activity. So when your metabolic activity at a tissue is up or down, that can alter how the blood flows to that tissue. And kind of like the big ways to sort of think about this is when there is increased metabolic activity at a tissue, what that normally would mean is that you are going to be using oxygen quickly and making more carbon dioxide. So you would have low oxygen levels in the tissues. I don't know why I didn't write that down there. And an increase in carbon dioxide. Oh, I know why. You might have more potassium there. You might have more hydrogen ions there because of the carbon dioxide, you know. And so any of those little changes, you know, decrease in oxygen concentration due to increase in metabolism, an increase in CO2 because of an increase in metabolism, a decrease in pH, you know, because of the carbon dioxide, or maybe the lactic acid if you've run out of oxygen. Those things tell when that accumulates locally, your intrinsically that tells your body that you need more blood there. You need more oxygen, you need to wash away that CO2, you need to get rid of the hydrogen ions and all of that. So that is an intrinsic type of regulation. It's driven by what's happening at the tissue. And that would cause a vasodilation because you want to increase blood flow to that area. If for some reason you're not using that tissue very much, how your body would know that is there'd be plenty of oxygen in that blood. It's not leaving the blood and moving into the tissues because you're not really needing it. And so that increase in oxygenation in that area would trigger a vasoconstriction. You don't need more blood there. All right, so this is oftentimes referred to as autoregulation, the way of the ability of the tissue, what's happening, you know, metabolically in the tissues can control the way blood is flowing. What else do I want to say? So on the previous slide, or at the slide a couple slides ago where I said regulation of blood flow, I mentioned paracrine regulation for a minute, and I said molecules in one tissue are produced and they affect other tissues in that same organ. An example of this would be kind of how we can see smooth muscle relaxation in the vasculature. So the inside layer of the blood vessel, the tunica interna, if you remember, oftentimes will produce signals that will influence the smooth muscle activity in the tunica media. So the signals produced by the inside of the tube will control the tension or the smooth muscle activity of the muscular layer. So there are things like, somebody asked me this in one of our review calls last week, what causes vasodilation? Nitric oxide is produced by the tunica cells of the tunica interna. Bradykinin is another thing that can do it. Some of the prostaglandins can do it, and they trigger vasodilation. That would be also considered intrinsic. There's another, in terms of vasoconstriction, there's a signaling molecule called endothelin-1, and that will stimulate contraction in those vessels. So that's what I was talking about with paracrine signaling. All right, and then there's this type of control called myogenic autoregulation, and all this is about is how the vascular smooth muscle responds to changes in arterial pressure. So what that means is if the pressure in the vessels change, another way to control it is myogenically. And so what that means essentially is if you have increased perfusion pressure, to perfuse means to bring more blood. So if more blood is going into a certain vessel, and as that blood goes in there, the pressure, the blood, as the increased blood flow goes into that vessel, the pressure in that vessel is going to go up. So that increased pressure would cause the arteriole to stretch a bit. Arterial wall, it's a tube, right? You put more fluid in there and higher pressure, it causes the vessel wall to stretch a little bit. When the vessel wall stretches a little bit, that little bit of stretch, these are like kind of like stretch sensitive receptors built in. That little bit of stretch will cause this like, reflexive contraction. This is an inherent property of most smooth muscles. Now, I will say that there's kind of a limit to that situation, and there's a window where the vessels will respond. If you go outside of that window, either too high or too low, the response is kind of null and void. But that's something you'll learn someplace else. So this is a way that the blood vessels themselves can kind of control the size of the tube based solely on stretch. And this is another type of what we call autoregulation. This, interestingly, is how we keep the pressure that flows into our brain constant. Your brain tissue, well, no tissue for that matter, but the vessels in your brain don't like huge changes in pressure. They don't want the pressure to go up and then down and then up and then down. We want to try to keep the pressure pretty consistent. And how we do this is we have, especially those arterioles, have the ability to myogenically autoregulate, to kind of tweak slightly just based on pressure or stretch alone, not on O2 or CO2 or all that other stuff that we talked about, just on stretch to try to keep that flow consistent. And again, that works really well as long as your blood pressure stays in the normal range. If your blood pressure goes too high, you lose the ability to autoregulate. If it goes too low, you lose the ability to autoregulate. But as long as you stay in the window, you're okay. And that's really helpful. And the very final thing I want to mention is this situation called hyperemia. Hyperemia just means like a lot of blood in an area. So if you put a lot of blood in an area, we call that hyperemic. And we can do this as a reaction to something or just as a byproduct of metabolic activity. So reactive hyperemia happens as a consequence of vasoconstriction in a tissue. So if we were to vasoconstrict a tissue, so the blood goes in and we vasoconstrict, what that will do ultimately is it will cause the downstream tissue to start to produce waste that can't be removed, right? Because the way blood flow is good for delivering nutrients, but just as effective at getting rid of waste products. So this is a way that we kind of trap, if you will, metabolic waste in an area. If enough metabolic waste accumulates, then that will ultimately cause vasodilation because enough CO2 will accumulate, enough hydrogen ion will move into that area, and that will override the constriction and it will cause vasodilation. We were just talking about that. So that's called reactive hyperemia. Active hyperemia is just what happens as a result of an increase in blood flow. So like when you're exercising, your cheeks turn red, sometimes your body kind of gets like reddish color, excuse me. That's just due to increased blood flow because of the increased metabolism. Both situations will cause the skin to turn reddish. So whether it's reddish because we've constricted the vessels, which is causing, this accumulation of waste. And sometimes that will happen. Again, the reactive hyperemia is a little bit confusing. The reactive hyperemia, you have to kind of think, remember, we're always shunting blood. Like you only have so much. And so we have to, move it where we need it. But if we don't need it in an area, we can constrict. And if you really constrict to an area, that's what causes this sort of reactive hyperemia, because you're just not getting the blood delivery and getting rid of the waste products. And then, just with a consequence of just overall metabolism and the increase in blood flow that we see as a result of that. All right, so that was a lot. Go back and revisit the variables. again, what you're going to focus on are equations and how to kind of figure out how blood flow is controlled, keeping at the forefront of your mind the role that the diameter of the vessels play. And that's the big one, the radius of the tube. That's the big variable, most importantly here, because it affects resistance, and because small changes in the radius are mean large changes in both resistance and blood flow. And of course, where we see the effects of changes in the diameter of the tube is at the arterioles primarily. That's the big one. Small arteries, but really the arterioles. All right, you guys, that is the end of this chapter, which is lecture 16 and corresponding to chapter 14 in your book. If you're reading the book, you'll know that there's a whole bunch more in this chapter for blood flow control, but we are out of time. We need to go into other things. So if you're interested, and it's kind of interesting, it goes into blood flow to the skeletal muscles, blood flow to the skin, blood flow to the heart, blood flow to the brain. It's the same ideas, and I think you can kind of probably think through it, but you're gonna you can read that on your own. We will be talking about blood pressure in the labs. So you're going to do a lab on blood flow and blood pressure. So you're going to get more detail on that in your labs if you haven't already had that. All right, I will talk to you soon.